I'm excited to be joined today by Dr. Jim Roney. He is a professor of psychological and brain sciences at the University of California, Santa Barbara, where he runs the Human Behavioral Endocrinology Lab. Jim, welcome to the Nature and Nurture podcast.
Thanks very much for having me.
We're just beginning to talk about off camera about hormones' role as coordinators in the body. What does that mean?
Yeah, so I think if I were to sum up in one word what hormones do, what they're for, the word I would use is coordination. And then I would say some more words. But I think this follows from the fact that it's one of the things that makes hormones really interesting is that they're released into the general circulation. And so as signals then, that means they can really calibrate mechanisms and parameter settings throughout the entire organism, right? They can calibrate settings in the body outside of the brain, but also simultaneously in brain mechanisms. And so if we're going to understand the functions of hormones, then I think the best way to do that is to first, so there's the coordination angle, but another big part of the argument that I'm trying to make in terms of how we should go about building theories about the functions of hormones is that we need to understand how they mediate input relationships and how those map into certain outputs. Maybe I could just start with a really concrete example of that to get a sense of what I mean. In seasonally breeding species, oftentimes in the breeding season in males, there's a very big seasonal effect on their testosterone concentrations, right? In the breeding season, testosterone goes up, and in the non-breeding season, it often falls to very low, even castrate levels. And so we can think of things like photoperiod that reliably indicates season as an input to testosterone that drives it up in the breeding season. And other things affect it as well, like the presence of fertile females from the same species can drive testosterone up as an input. Right, so we've got these inputs to testosterone causing it to go up. And then when females aren't present or it's the winter. And then on the other side of the equation, we've got this whole set of coordinated output effects that testosterone has. Right, so it's causing the growth of secondary sex traits, like in some species of deer, for instance, antlers will grow under the influence of testosterone in the breeding season, but they fall off in the winter when testosterone drops. Muscle mass increases. So we're getting these effects in the body outside of the brain, but at the same time, testosterone is circulating to the brain and affecting things like sexual motivation, aggressiveness, and willingness to compete with other males. And then because all of these behavioral and other effects cost energy, it's thought that testosterone has other effects that grab that energy when it's needed. So like suppressing immune responses or suppressing fat storage and taking that energy and putting it into muscle mass and antlers. And so we get this whole kind of coordination of different things that are going on simultaneously, both in the brain and the body, affecting both behavior and these physical effects. But we can understand their function if we understand the input cues that are driving testosterone up. So this all makes sense as a coordinated strategy to compete for mates when mates are available in the breeding season, right? But when they're not available in the winter, then these would all just be costs that might harm survival. So testosterone drops. And then other inputs to testosterone then that make sense given those outputs, things like food shortage will cause testosterone to drop, illness, right? You don't want to suppress immune responses if you're sick. And so what I'm arguing for then, what I call this theoretical framework approach, is to understand the functions of hormones, we have to understand kind of the list of inputs that drive hormones up and down, and then the list of coordinated outputs. And if we can see that the coordinated outputs are a functional response to the inputs, then we can build functional theories about what the hormones are doing. So that's the approach that I'm trying to promote. And it's a very kind of common sense way of thinking about it, I think, on the one hand. But on the other hand, I don't think that approach is that common, especially in psychology. I think it's a little more common in biological anthropology. But in psychology, there's a kind of a different approach that's often taken that I refer to as a parsimony-based approach, where it seems like the goal is to come up with the one main thing. And so in the testosterone example in humans, there have been debates about whether it's an aggression hormone, or maybe it's a dominance hormone. Aggression is too specific. And then others have argued, no, it's really about promoting social status. And it's not that that's empirically false. It's just that it doesn't really explain the functions of the hormone. It doesn't tell us what the inputs are to cause testosterone to go up and down. And it doesn't explain why testosterone would be a good signal to affect something like pursuit of social status. And it neglects all of the other coordinated effects that it has outside of the brain. And so what I've been trying to argue for is, this kind of input-output mapping, coordinating function approach is going to give us more purchase on the functions of hormones than just trying to develop theories of the one thing that a hormone does.
It's interesting thinking about the different levels of functional analysis you can apply here. So one type of input might be very mechanistically, like on the cellular level, you're thinking, what does testosterone do? The hormone actually binds to this receptor and it triggers this whole biological cascade of events that leads to growing the muscle or growing the antlers or whatever. But that's atheoretical. You don't really understand the why. And then conversely, if you create this grand ultimate evolutionary theory of what is the sex hormone doing, it's essentially improving fitness or dominance or aggression, like all of these things that are going to, in the end, lead towards more reproductive fitness. But you can't actually explain all of the middle stuff between how you get from that one theory of what testosterone is doing to the actual endpoint of how does it technically improve fitness or improve the likelihood that you leave offspring. Then you're missing all of those inputs. And I find myself bouncing between those two. I generally understand less of the proximate biological details as a psychologist, but I'm very interested in those big evolutionary grand stories telling you like what the adaptive purpose of this hormone is.
Yeah, good. Me too. And yeah, I think you really well identified these kind of complementary levels of analysis. And sometimes in biology, they refer to it as proximate and ultimate levels of analysis. So there's a story about the selection pressures and the functions. And I should say, when I talk about function, I'm really talking about and fitness and reproductive success. It's really referring to reproductive success in ancestors and in ancestral environments, right? There's no guarantee that the effects that are occurring today would also be promoting reproductive success in the same way, right? So we're explaining current design based on past selection pressures. And one of the things that's interesting to me about hormones is that in humans, a lot of their functions seem to be pretty well conserved from non-human species, right? So it's sometimes easier to think about function in non-human species, as in the example of seasonally breeding species, where the function of this testosterone, suite of testosterone effects, I think is pretty clear. But then when we see parallel patterns in humans, it really suggests that we had conservation, some of these mechanisms from non-human species, or something that biologists call homology. And so one of my main strategies in trying to study human hormones has been to read the non-human literature really carefully for clues, because this is design that we're descended from. We expect there to be some modifications of that design in humans, but it gives us a great baseline starting point for generating hypotheses about how the hormonal mechanisms will work.
Yeah, in the first year of my PhD, I probably read more animal research than humans, which is a bit surprising being in a psych department, or a lot of it was like yours, these sort of evolutionary psychology / biology theoretical reviews that are relying on a lot of animal research to build the theory and then arguing how the same thing applies in humans.
Yeah, I had the same exact strategy when I was in graduate school. When I first started at the University of Chicago, I was in a weird situation where my original advisor retired after 1 year. And I was just, what do I do? I was on my own. And I spent a lot of time in the library, and I was interested in mating psychology in humans. But a lot of that early research had just relied on kind of self-report surveys, which in a sense don't really get at the mechanisms in real time or in an ecological context. And so one of the things I started reading about was in non-human species, what happens when they encounter potential mates. One of the findings I kept running into was, especially in males in vertebrate species and sexually reproducing species, when males encountered potential mates, they would show these rapid increases in testosterone. And they didn't require physical contact. So I realized this could be tested in humans. If we just stage social interactions between men and women, we could see whether that same testosterone response occurred. And so that kind of, by reading the non-human literature, that kind of brought me into hormone research. And then eventually I branched out into looking at ovarian hormones and so on. But yeah, I think it's an underutilized source of clues and evidence, especially when we're talking about things like motivational systems that we might expect to be more conserved. If it's something like language, then yeah, maybe that's more uniquely human. But yeah, for motivated behaviors, I think the non-human literature is underutilized. And
I still don't understand much of the biological details of how the hormones are produced and transmitted through the body, but I remember reading that for some types of responses, it's still a mystery how rapid acting they are. Something you might see is your testosterone levels go up just a tiny bit after the team scores a goal or after you see some attractive mate enter the room. And from what I remember reading, it's like most of the hormone signals that we really well understand happen over the course of like many minutes or even hours. And yet you see these very immediate responses and we're not fully sure how it works. Is that still true?
yeah, I think that is true. So there are two different timescale issues there that are important. So one would be the time lag between the stimulus event and when you're seeing a hormone increase. And yeah, so the HPG acts as signal cascade. There's conflicting evidence about how long that should take before you see a response. And if you dig in that literature, you can find some, so that could be anywhere from 15 minutes to 30 minutes or so for that whole cascade, go from hypothalamus to pituitary to the gonads and then release a hormone like testosterone. But if you dig in the literature, you'll find these like interesting little findings that suggest things like lactic acid can have effects directly on the testes and cause an almost immediate release of testosterone. And so there are other potential mechanisms and routes aside from kind of the classic slower acting routes that might cause faster release. But then the second time lag is how long does it take a hormone to do something, right? To actually have some effect. And so you may have been implying there, the kind of the classic mechanism of action is by affecting gene transcription, right? To binding to these receptors that go into the nucleus, cause certain genes to be expressed or inhibited. And that takes like hours or days for those sorts of changes to manifest themselves. But again, also in the literature are other findings suggesting that there are some membrane based receptors that act much faster. And so there are a number of animal experiments showing that you can administer testosterone and see some downstream behavioral effects within 15 to 30 minutes. And so those are probably not through the classic gene expression pathway. Those are probably through fast acting pathways. And so the gene expression pathway is very well established. It's slower. We know it's there, but there's also data suggesting that there are these faster acting pathways as well.
Is understanding these more cellular level mechanisms something that has just come through osmosis in decades of doing this research? Or is it something that, as a psychologist and behavioral endocrinologist, you've practically needed to know and implement in your research?
Yeah, it's a good question. I think it's more osmosis, actually. So yeah, because I don't work with animals and actually get in there and work with the receptors and so on. So it's been more about interpreting results and then theorizing about mechanisms and the timescales at which the mechanisms might be working. So in those cases, it's consulting with what the neuroscientists are doing to get clues about that. But yeah, I'm not in a position to do that research myself. So yeah, I think you can still draw some interesting functional inferences about what the hormones are doing without getting down to that cellular level yourself. But sometimes yeah, you need to know that it's plausible that the mechanism you're proposing could actually work in the time scale you're proposing. So going into the neuroscience literature can help you see whether that is possible.
When I first started getting interested in cognitive science, it was largely through evolutionary psychology. I liked thinking of these big picture ideas, but as I'm sure it gets criticized a lot as like sort of armchair theorizing. You can come up with an evolutionary story for everything, even if it ends up, in fact, being a spandrel. And I think in retrospect, what got me really interested in hormones, maybe this is the same for you, is that it combines the theory with a really neat mechanism. And especially when you're looking at mating behavior, which seems to be what these ultimate evolutionary theories generally all boil down to, if this is something you can so directly tie to sex hormones.
Yeah, I agree. So it's what I was suggesting before, that I wanted to know more about mechanisms, and the hormones seem to be closer to the mechanisms in some way, right? Relative to something like a self-report survey. Yeah. And another interesting thing to me, again, is the similarity across species, because what that suggests to me is that even though there have been lots of changes with speciation events, lots of changes to the genetic background, obviously environmental changes and social changes when you go from one species to another, if you see hormonal mechanism working in very functionally similar ways across species, that seems to suggest that there is some conserved design for producing specific functional effects. And so it becomes so, right. In human psychology, right, we have various alternative explanations for kind of psychological outcomes where maybe there's something about human environments and social learning and cultural change that's driving psychological outcomes, as opposed to them being generated by a mechanism that was designed by natural selection for that specific purpose. But when you see a mechanism working in the same functional way that it's working in other non-human species, those non-human species don't have all of these human specific inputs to their development, right? And so that conservation suggests that there's, at least to me, that there's something specialized about the mechanism that's producing that same sort of functional outcome. And so a concrete example that I think would be maybe less controversial than things like mating behavior, or people have strong feelings about it, would be some of the signals that go into satiety and hunger in humans, right? We can understand their functionality very clearly. Many of those signals are highly conserved from non-human mammals, suggesting that it's not some kind of human-specific developmental pathway, right? It's a conserved biological pathway. But it's also interesting that we're talking about psychology still, right? Because feeling hungry is a psychological outcome. And yet it's pretty non-controversial, I think, that we have specialized design, specialized adaptations that are regulating our satiety. I like to draw that analogy for mating psychology, where it's a little more controversial, right? Because people want to argue that no, mating psychology is a culturally learned, culturally specific process. But if we can have psychological mechanisms that regulate our hunger and satiety in a way that's conserved from non-human mammals, I think there's a very natural analogous argument that can be made about attraction, courtship, and sexual desire, and so on.
When thinking about the evolution and conservation of these hormones, it seems like early in their evolutionary history, it always starts as hormones as coordinators of something very physiological. And then the way I've been thinking about it is that over time, as the species become more complex, more intelligent, more social, the hormones take on more psychological functions. And the two main ones that I learned about are testosterone and oxytocin. And with both of those, testosterone is obvious in the sense that it's a primary sex hormone. So long before we knew much about the psychological effects, you can easily point to it and say that testosterone is going to be what leads to the primary and secondary sexual characteristics being developed. And then for oxytocin, I learned about how before we considered it this love hormone, people studied it in a medical context as the hormone that triggered the muscle contractions during birth and during breastfeeding. And then it seems like whatever the physiological function of that hormone is, it becomes psychologized. So it makes sense that the sex hormone then influences sex drive. And it makes sense that the hormone involved in the physical parts of maternal care then become involved in the psychology of care.
Yeah, I think that's a great insight. And I think it's really interesting to think about the phylogeny and the sequence of how these kind of coordinated outputs all come together. And so, yeah, I have the same sort of thought. So For something like testosterone, if it's regulating sperm production, which it is in sexually reproducing species in males, then anything that you want to coordinate with fertility being higher. And so like again, in seasonally breeding species, in the winter, fertility is also shutting off. Testosterone is a natural signal to do that, right? To have that coordination. And I think you're exactly right with oxytocin as well, that you would expect that other things. But I think one of the things I'm trying to argue for is that to understand these hormones, we really need to understand the inputs and the context in which they're changing, with it going up and down. And so birth and lactation are important input contexts that can help us understand the downstream coordinated effects of a hormone like oxytocin. But one of the frustrating things about the hormone literature to me and these parsimony-based theories is that oftentimes they focused, or a research literature will come to focus on only inputs or only outputs. And so in the
Mhm.
case of oxytocin, I think because of this intranasal administration research that took off for a while, the focus was all on outputs, right? It was all on, if we jam this signal into people's noses, um, what's going to happen to them? What can we measure? And then how can we develop a theory that kind of unifies all of these effects, seemed to be the goal. But in doing that, they were completely neglecting what are the natural inputs that are going to cause oxytocin to go up or down. And yeah, in the case of something like lactation, there's this idea that people had argued that there's this oxytocin paradox, in the sense that when they administer it, it tends to have a lot of pro-social effects, but every now and then you see some things that look antisocial, less trust of out groups, or more discrimination against out groups, or things like maternal aggression in,
Mhm. The mama
in
bear
rodents.
effect.
Yeah, exactly. And so this is a paradox. How can we have both pro-social and antisocial effects? But it's only a paradox if you're trying to develop a parsimonious, unified theory to say this is the one thing that the hormone does. But if you're linking inputs to outputs, then during lactation, it makes perfect sense that you have not only the obvious things like promoting milk letdown, which it does directly, and a lot of pro-social things like recognition of offspring and priming of maternal behaviors, which you can show experimentally oxytocin has effects on. But it also, there's no paradox in understanding that it would increase maternal aggression toward intruders. So female rodents who would usually retreat from an intruder, if they have pups, no, they attack. And nice experiments show that oxytocin is causal in priming attack. But once you link it to the outputs circumstance, I'm sorry, the input circumstance, so there's no paradox anymore, right? You understand that maternal aggression is perfectly well coordinated with these other behaviors, but also with a whole series of physiological effects on insulin, effects on basically mobilizing glucose and shunting it into milk production. All of the effects just come together nicely. And what we might need to do then is to figure out what are the natural input contexts that cause oxytocin to go up or down. Can we understand coordinated outputs when we understand the circumstances in which it's changing?
I did a podcast with Sue Carter earlier this year, and we were talking about that. And she was convinced that many of these seemingly paradoxical oxytocin effects are actually confounded by vasopressin.
Yeah, that brings up another issue, another kind of complication to the approach that I'm taking, although I think it makes it really interesting, which is that usually in an input circumstance, something that's driving one hormone up, that's not the only thing it's affecting, right? You'll have multiple signals that are all being affected at the same time. So multiple hormones, but other transmitters and so on. And so those might combine in various ways to affect outputs in different ways. And so in principle, then what you have then is maybe codes for certain adaptive input circumstances that involve multiple signals. When you get different subsets of those signals responding, you get different subsets of outputs as well. And so elevated oxytocin in the presence of elevated vasopressin and cortisol might be producing different outputs than elevated oxytocin in the absence of those signals. And again, that's one of the problems with just an administration study is you might be increasing oxytocin, but under natural circumstances, it would have been combining with other signals that are absent in that manipulation. So that makes the story a lot more complicated, I think, but also potentially more interesting when you're looking at how these signals might combine. It adds a lot of new.
It's to make the input even more complicated and interesting. This is something I've still been struggling to understand. I think you've written about it in some of your testosterone reviews, which is that beyond the effects of the actual circulating hormone levels, there's going to be tremendous individual variability in how the receptors work. So like someone could be more or less sensitive to testosterone, so that the person with lower testosterone, if they have this higher sensitivity, might actually have a greater effect, or vice versa.
Yeah, yeah, that's another complication. Things are complicated.
Yeah.
So for testosterone in particular, the androgen receptor gene has some known polymorphisms that are interesting in that they're continuous. So the number of CAG repeats in the androgen receptor gene has a pretty normal distribution. So it's not just there are two different alleles, it's this repeat polymorphism. And there is pretty good physiological evidence that individuals with shorter numbers of repeats have larger physiological effects to the same amount of testosterone. So if you administer testosterone and measure, say, metabolic responses to that, it's larger if they have shorter repeats. And when we're thinking about individual differences, it's possible that the same amount of testosterone would be affecting two individuals differently, in this case, related to a genetic polymorphism. But yeah, I mean, there could also be developmental, environmental calibrators of individual differences as well.
Is there a simple explanation other than just memorizing the directionality to why it makes sense that shorter CAG repeats would be associated with more sensitivity to testosterone rather than the other way around?
Oh, yeah, I don't really know the 3D structure of the protein. So the number of repeats is going to basically affect how the receptor protein is going to fold into some 3D structure. And then that is going to have the downstream effects. So why shorter number of repeats leads to more sensitive? Yeah, that at that biochemical level, someone may, but I don't know the answer to that.
Another place where I saw you write about a role that this androgen receptor number of CAG repeats could play a role is on the effects of prenatal testosterone on really a whole bunch of different outcomes. But I remember one that is, see, there seem to be mixed opinions about it, is prenatal testosterone's role on determining sexualities. From what I understand, there's evidence that in females, higher prenatal testosterone is associated with an increased likelihood that the child will grow up to be a lesbian, but you don't see the same thing in males. You don't see lower testosterone or higher estrogen being linked to sexuality.
Yeah, generally the theorizing about organizational effects for hormones on sexual orientation, it's applied to both males and females, right? So the idea
Mhm.
here is that it might not necessarily be maybe like fetal testosterone hasn't been measured in a way that's predicted male same-sex attraction. But the theory is still that androgen signaling is crucial for developing so attraction to women. Yeah. So are there specific studies that you have in mind there that for that?
Before I started my PhD, I did research with Megan Herting, who studies hormones and brain development. And my master's thesis was on congenital adrenal hyperplasia, which is this intersex disorder where females are exposed to heightened levels of prenatal testosterone. And you see a whole bunch of interesting effects. We were looking at brain differences and emotion processing differences. But I remember in reading about this that girls with CAH are much more likely to grow up to be gay. And I thought, oh, that's interesting. So it must have to do with the prenatal androgens. But then looking into the same thing in males, I don't remember seeing any consistent evidence one way or the other. Because if you take that starting place of, okay, more testosterone in females makes their sexual attraction more male, then maybe you would think that lower testosterone in males or higher estrogen in males has the opposite effect. But I didn't find that.
There are some conditions, I guess it depends on how you define male, female, but if the XY karyotype, so you have people who are genetically male. But if they have a condition called congenital androgen insensitivity syndrome, so basically their androgen receptors don't work, then they tend to develop a female phenotype, which includes attraction to men in most cases for the individuals who have been surveyed. And so that is a case where essentially, because the androgen receptor isn't working, it's like having really low testosterone, and it's associated with attraction to men despite having XY chromosomes. And I think that's part of the argument for the hormone hypothesis for sexual orientation and it applying to men as well as women. I
I
mean, in
learned
that
that
case, go ahead.
in Carol Lubin's testosterone book. I thought that was so cool because it's not just the low testosterone, so they're more feminine. It's all androgens. So they don't even develop body hair or body odor or any of the pre-puberty sex, non-specific secondary sex characteristics.
There is some conversion to estrogen, the available androgens. So you do get some estrogen effects. But right, I think anything that's operating through the androgen receptor is not
Yeah, that
having
was cool to
effects.
find out too, because originally I thought, okay, so they don't have the testosterone necessary to go through male development, but they don't have ovaries either. So maybe they're just going to be destined to be like a girl for life because they won't have the necessary resources for female puberty. But it seems that the amount of estrogen that just testes produce is enough to trigger like breast development and fat redistribution around the hips and all that stuff.
Yeah, that's right. It's not just the amount of testosterone, not the amount of estrogens being produced, but the fact that usually some of that testosterone is interacting with the androgen receptor, but because it's not, in this case, more of it may be converted into estrogen through the aromatase enzyme that both
Mhm.
males and females have. Yeah, I wouldn't say I'm an expert on that condition, so I don't want to dig too deep into it, but you're right. It does seem like there is enough conversion to estrogen to have what appears to be a more female typical puberty.
Have you heard about those Russian foxes breeding experiments?
Is this
It'll be related to hormones in a roundabout way. I'll get there. So there were these experiments looking at a control group that would just breed normally, no selective breeding. And then there was one group where they went for the most docile foxes every single generation, and one
okay?
group where they picked the most aggressive ones. And after many generations, the docile ones not only acted like dogs and they were super friendly and would lick humans, but they started looking like dogs too. Like they would get floppy ears and spotty coats and softer fur. And the more aggressive ones would have physiological changes as well. They would have, I don't know, bigger canines or rougher fur. They would look more wolf-like and more aggressive. So the idea there is that whatever genes are associated with the psychological traits, maybe it's not a genetic effect. It could have been something like it could have been hormones, prenatal hormones that were being genetically selected for, but it was like hormones driving both the psychological and physiological traits. I learned about that study. And then a while later, I heard about this controversial study at Stanford where they developed this gaydar. It was an AI facial recognition algorithm that could predict well above chance whether someone was gay or not. And they did a whole bunch of controls like the images were black and white and they cropped out the hair and they cropped out the outfit. So it was literally just facial features and nothing like maybe they had different style or something that could confound it. And I was thinking that, okay, in the same way that these Russian foxes might have changes to their fur or their teeth or ears or whatever that are associated with behavioral traits, maybe there are physiological traits that are subtly associated with sexuality. So even if no human could detect it, maybe what was happening is the AI was looking at like subtle differences in like cheekbone structure or something like that might be correlated with sexuality.
Yeah, I'm not familiar with that study. It sounds interesting. Yeah, it's a possibility, but I do think one of the interesting things about sexual orientation in general that is, it's mosaic in a sense, right? In that at least from my human casual intuitive impression is that people who have same-sex attraction oftentimes are otherwise sex typical in a lot of ways, right? In terms of their morphology, this ability to have this change in whom they're attracted to while maintaining otherwise sex typicality. One of the interesting things about it, and even things like what's called sociosexuality, so openness to uncommitted sex or having multiple partners, the available data that I've seen suggests that for the most part, gay men are similar to straight men, sociosexuality. So there's this element of sexual psychology that's sex typical, but the attraction is sex atypical. And to me, from a mechanistic perspective, that suggests that something during development, probably having to do with androgenization through some pathway, is affecting some mechanisms, but not others. Whereas what you're saying is maybe in a more subtle way, there's a broader fact that's generalizing the morphology, at least as detected by the AI. I think it's unresolved, right? Just how general the changes might be. We know some people are working on that. There's some people here at UCSB who are trying to do a project on try to increase the amount of knowledge about people who don't have opposite sex attraction, so bisexual, gay, looking at mate preference patterns, right? Are they similar to uh, their same sex, or are they similar to the opposite sex, and so on? I think there's a lot that's unresolved there. Need some better data. So, yeah, and that's a really interesting study. But certainly for things like, and sometimes this has been explained by maybe the hormones are having different effects at different times of development. But certainly the overall physical morphology is sex typical, right? They still have, we recognize male genitalia, female genitalia. There might be small changes in,
I was thinking of subtle changes like those 2D to 4D ratio studies, like finger length ratios are associated with prenatal testosterone, and then the prenatal testosterone is associated with a whole bunch of later life outcomes. So it's not like the physical subtle difference in your finger length is causing anything, but they share this common root that ends up being predictive of a whole bunch of things. So again, in like those very subtle physiological differences, even if the grand morphology is the same, you might see that linked to really any personality differences, not just sexuality.
sure, yeah, right. When you're getting to that level of subtlety, I think it's an empirical question. I'll have to look up the AI study. That's a published.
Yeah,
Yeah,
it was definitely controversial. I don't know about the methods, but more just because you can, why did you do it? Like, why would you train the AI to be a gaydar? But another question about these continuous effects of hormones is really how continuous are they? Because I've seen for some people have this intuition of more testosterone means more aggression, more sex drive. And maybe they assume like some linear relationship or at least some straightforward relationship. But I've seen other evidence that the effects are more permissive or categorical. And it's like within a fairly broad range, you can change the hormone and the behavior doesn't really change. And it's only if you fall below some critical threshold that sex drive starts to go down, or if you go above some critical threshold that aggression starts to go up in a measurable way.
yeah, I think that's exactly right, especially for men's sex drive, right? It seems to be more of a threshold effect for testosterone. I think things are a little bit different in women with ovarian hormones predicting their sexual motivation. And actually, we're getting ready to submit a paper right now on exactly that question about continuous variation in testosterone in men and how it might relate to their sex drive and sexual motivation. So it is the case, we know from some earlier studies, that if you chemically block testosterone production, so you make men's testosterone go to very low castrate levels temporarily, that does have a very clear measurable effect on their sexual motivation. So their rates of fantasy, self-reported desire, behaviors all go way down. But what was interesting in those early studies is that you could replace testosterone at about half of the average baseline levels and restore those variables to basically their pre-manipulation levels, right? Suggesting that men were making more testosterone than they needed to have just typical sexual motivation. And so what we did in the study that we're getting ready to publish is we actually followed men for 30 days and got saliva samples each day across the 30 days. And then we had them fill out a daily diary and they indicated their desire for sex on each day, as well as a bunch of other things. And we see no within subject correlation between fluctuations in testosterone and fluctuations in sexual desire, which is consistent with that threshold effect, right? Because none of them are hypogonadal. So they should all be in the physiological range for testosterone. And so if there is a threshold effect, then you don't really expect those fluctuations to predict desire. And that's what we found. But I think you're right. There is this common intuition that, oh, yeah, testosterone, if you have a high testosterone day, then you're going to have a higher desire that day. And we don't see evidence for it. I think it's the first study that's tracked it from day to day. People have measured, say, across a week or monthly measures and so on, and not seeing a correlation either within them. So, yeah, so I do think it's a little less continuous there. Whereas in women, what we're finding is a more continuous relationship with estradiol and progesterone, especially with progesterone, where on days when progesterone is higher, women are self-reporting that their desire is lower. So I don't know if you want to get into that.
So yeah, let's get into that for sure. Because so in my lab, we're studying all the main pubertal hormones of testosterone, estrogen, progesterone, and DHEA. And I really only have a good theoretical sense of what testosterone is doing. And I've started to learn more about ovarian hormones. So maybe I can explain what I know and you can fill in what I get wrong. From what I understand, estradiol peaks just before ovulation and it's positively associated with sex drive. And that kind of makes sense given the timeline, because right before ovulation is when you want to have sex. You want to have offspring, like from this evolutionary incentive way. And then sex drive dips, I think, to lowest levels right after ovulation. And that's when progesterone is at highest. So there's a negative relationship between progesterone and sex drive. And then some of your work that I read showed that progesterone is negatively related to sex motivation, but positively to food motivation. So this might explain like period cravings, or if you do get pregnant and you're not going to have a period, then maybe like you're storing energy for the pregnancy. So either way, it makes sense why food drive would go up and sex drive would go down after ovulation.
Yeah, so that's an accurate depiction of the patterns that we've seen in our daily diaries. Yeah, surprisingly, I think we had the first study that was measuring every day across the cycle of desire and then hormones on most days in 2013. Even though it seems like a pretty basic question, right? What do estrogen and progesterone do or how are they associated with women's sexual motivation? But yeah, we see exactly what you had described there. So, right, just before ovulation is when estradiol is peaking, and it's actually mechanistically involved in triggering ovulation, which makes it a reliable signal of impending ovulation. And so in humans, there's a period of time called the fertile window, which is the only time that sex can produce conception. And it runs from about the day of ovulation to about 5 days before. So that's a period of time when estradiol is peaking, but progesterone is still low. And then after the fertile window, the second half of the cycle called the luteal phase, conception's not possible at all. And then progesterone goes very high. And so what I've theorized is that estradiol and progesterone act as a really simple kind of two signal code that oppose each other, right? So for sexual motivation, as you were describing, estradiol is turning it up and progesterone is inhibitory. And so given the way that the hormones are shifting across the cycle, you would expect peak in the fertile window. Then progesterone kind of shuts that off once the opportunity for conception is passed. And so in a lot of non-human species, females are only sexually receptive in their species-specific fertile window. And the two hormones having those opposing effects is a way of kind of turning on receptivity and then turning it back off. Humans are not as, sexual receptivity extends across the entire cycle. But what we're finding in our data is that the hormone influences are still acting in a similar way, just more subtle than what we see in non-human species. But then what you alluded to is, in terms of food intake, it's the mirror image of what's described for sexual motivation. So not just progesterone being positively associated with food intake, but estradiol was negatively associated with it. So there's actually a sharp drop in food intake in the fertile window. that we found was mediated by daily measures of estradiol. And then as progesterone rises in the second half of the cycle, women's daily self-reported food intake was rising and tracking the rise of progesterone.
But I
And
remember
so,
reading about that negative effect of estradiol and food intake, seeing myself very quickly come up with and then dismiss like an evolutionary psychology story that doesn't actually make sense. So the idea here is that at least right now in Western culture, the ideal female body type in the middle in the media is like very skinny. And then maybe the thought was, okay, sex drive is going up. So food drive will go down because it'll make you look extra skinny, extra hot, according to these beauty standards. And then I was thinking, but that does seem to be more of a cultural effect. And I know in some other cultures or across history, a larger body type was seen as more attractive. And just evolutionarily, that doesn't seem to make much sense. Like, I don't think other animals have the same sort of attraction where it's like they're going to be going after the skinnier female chimps.
exactly. And I think that's a great example where the comparisons with non-human species are really important. In female rhesus macaques, if you inject them with estradiol, their food intake goes down. They're starting from an overrectomized state. Then inject them with progesterone, it goes back up. And there's not the same motivation there, but the mechanism in terms of wanting to be thin for cultural standards, but the mechanism seemed to operate in a very similar way in humans. And to me, that suggests we're looking at a conserved hormonal mechanism that's affecting the motivation. And the similarity to non-human species is important evidence for what's happening there and you talked about. Our explanation there is, and we're borrowing from someone named Dan Fessler at UCLA, who first reviewed some of this evidence on cycle phase shifts in food intake. And his argument was basically that if you want to increase the motivation for one type of behavior, one way to do that is to inhibit motivation for competing types of behaviors. And so if there's increased sexual motivation in the fertile window, the time when conception is possible, you might see inhibition of competing motivations like food intake. So that's the idea that we're working with
Do you see
there.
testosterone effects on food drive or sex drive or anything else in females?
In our daily diary studies, we're not seeing significant effects of testosterone. And that is, again, it's contrary to this kind of intuitive belief and more widespread belief that testosterone is regulating libido, both men and women. So yeah, we don't see a significant effect. There is some evidence in hormone replacement therapy that really high supraphysiological doses of testosterone can increase and improve women's sexual desire, say in menopausal women. But someone named Kim Wallen at Emory University reviewed that literature carefully, and the conclusion that he came to was that estradiol is effective in increasing sexual motivation in hormone replacement, but if it's at pre-ovulatory concentrations, so it has to be pretty high, but still in the physiological range. Whereas testosterone is only showing effectiveness if it's at a supraphysiological level, so beyond what women normally produce. And so in a sense, that's potentially consistent with our data because all the women are within the physiological range, and there we're not seeing testosterone predict their desire.
And from what I understand, male estradiol and progesterone levels are more or less the same as female levels during the luteal phase when it's at its lowest and pretty stable. Do you know of any studies, whether in humans or animals, what happens if you give males much higher levels of estrogen or progesterone, similar to the increases that females see at the peaks of the cycle?
Let's see, I'm trying to remember. There have been a couple of studies where they've tried to tease apart the role of estradiol in men. Having trouble pulling up and spot their exact findings. I think the gist of their conclusion was that I think after blocking gonadal hormone production, if they were able to add back androgens with a chemical that blocks their conversion to estrogen versus not blocking the conversion to estrogen, that they were better able to restore men's sexual desire if they allowed testosterone to also be converted to estrogen. I hope I got that. But their conclusion was that there is some positive effect of estradiol on men's sexual motivation, maybe synergistically with testosterone.
Mhm.
But yeah, that's the most details I can pull up on that at the moment.
But why does it make sense to have both estradiol and progesterone have opposite effects when it seems like you could just have one and then have the whole input output function be like, if it's high, do this, if it's low, do this? Why do you need two that counteract each other?
Yeah, in the human menstrual cycle, estradiol is still being produced at pretty high levels in the luteal phase. And so I think, so we could back up a little bit and think about why the hormones have certain effects on the brain. And it goes back to coordination again. The hormones have primary jobs in the reproductive tract, right? So estradiol is causing the growth of tissue in the endometrium in case conception occurs, and there's creating this environment for an embryo to attach. And then progesterone is a very important signal for the secretory properties of the endometrium to make it a nutritive environment. But estradiol is still being produced at fairly high levels in the luteal phase. In fact, higher than most of the first half of the cycle, except right before ovulation. And so if you had a situation where it was just estradiol being the signal, then it's still pretty high in the luteal phase, and you wouldn't get, if you have progesterone reverse its effects, then you can bring sexual motivation down much faster. On the other hand, progesterone can't be the only signal because it's low throughout the follicular phase, and then it's rising only in the phase. So the way I look at it is these hormones have their jobs in the reproductive tract. They're getting the endometrium, the lining of the uterus, ready for implantation and making it a hospitable environment. But then they're circulating through the circulation up to the brain, and the brain is reading those fluctuations as information about things like, can I conceive right now? Is this a time that an embryo is going to be attaching? And then calibrating motivation to that information. But yeah, having two signals allows this much more efficient increase followed by just this sharp decrease when you have one on signal, one off signal, given the way the hormones fluctuate across the cycle. And I think they fluctuate that way for other reasons, for effects they're having in the reproductive tract.
And what is the function of estradiol and progesterone in males or testosterone in females if it's not directly linked to reproduction in the same way?
Yeah, that's a more complicated question. Presumably, right, there are also kind of synergistic effects through different receptors, and why it's designed that way. Yeah, I don't at the moment have a theory for that.
So in my lab, we're studying reward sensitivity in the brain and linking it to these sex hormones. And again, the one I know most about is testosterone, and testosterone increases dopamine production in the brain upon reward receipt. But the way that you normally measure reward activity in fMRI studies is through monetary games. So often there's like a gambling game where you can win or lose money, and you're looking at differences in brain signals in a win or a loss condition. And we're pretty much finding null effects for estradiol and progesterone. And once I read your work, I was thinking maybe the story isn't that they're not involved in the reward circuitry, but maybe it's that testosterone through this dopamine function is so generalized that you can find an effect from something as abstract as a monetary reward. But maybe estradiol and progesterone are so context specific to something like food and sex that we're just not able to pick up on it during our task.
Yeah, that's a really interesting thought, and I had similar thoughts about the sex versus food trade-off, right? Because they're both rewarding, and so somehow these estrogen and progesterone effects are domain specific enough that they're changing which is most. They seem to be changing which is most rewarding. It's not entirely clear that it's all a function of reward systems in that case. So for food intake, part of the effect might be through satiety mechanisms. So it might be that food intake is just as rewarding, but this sensation of fullness happens faster. So estradiol in non-human species potentiates some signals, satiety signals like CK. And so, yeah, it's possible that maybe that's not so much about domain specific reward modulation as it is affecting something like satiety. But yeah, that's something that I haven't really worked out or delved into the dopamine literature enough to have a specific theory about that yet, but it is a mystery to me. And you put your finger on it there. There doesn't necessarily seem to be just general reward systems, but something a little more domain specific. And that makes functional sense, right? Some things should be more rewarding under some circumstances than others. And it might not be the right approach to just think of reward in a very domain general way, which begs questions about using monetary rewards as your measure in all cases, right? Because, yeah, there are some papers I know that I can't remember the measure they use now, but they were looking at, it might have been just looking time or how much you would press a button to keep something on a screen. But they were looking at things like how rewarding various visual stimuli were across the cycle. And there may have even been studies that were, let's say, so sexual stimuli were definitely one. There may have been like infant stimuli and they're attracted, their
Mm.
reward
Oh,
value
I've seen those if it was related
Okay,
to oxytocin at all for the infant stimulus. And then I think testosterone for the sexual stimulus.
that could be. So, right. So somehow reward has to be more domain specific to have shifts in that way. And, what the mechanistic explanation for that is. Yeah, I'm not sure.
That was a cool example where I saw myself as more methodologically critical. I don't know if you noticed this with your grad students, but at the beginning, I feel like I was just taught the methods and go along with it and maybe don't know enough to critique them. So if I'm offering any critiques, it's more going to be on the interpretation side. But as I've further advanced in my research career, the critiques start to become more methodological.
Yeah, no, I think that makes sense.
Now, on the domain specificity of different reward drives predicting behavior, there's another big theoretical framework for risk taking and impulsivity, which is another thing that we study in my lab. And it's basically like reward and control processes having opposite effects in the same way that estradiol and progesterone, like one is inhibiting sex drive while the other one isn't, or food drive. So the idea is that impulsivity, and males tend to be more impulsive, especially during puberty, can be predicted by heightened reward sensitivity, holding cognitive control constant. But holding reward constant, it can also be predicted by lower inhibition. And the question, is it that males are more impulsive just because they're more reward motivated? And maybe you can connect that to testosterone and dopamine. Or is it also that they're worse at inhibition? And there's plenty of research showing that females, especially during the teenage years, are better inhibitory control. But even then, you don't know, is that just because females tend to mature faster in general, and then the males are going to catch up? Or could it be something unique to female sex hormones? So I was looking into that question, and there seemed to be mixed research from animal studies that maybe ovarian hormones have some association with inhibitory control, but that's hard to measure or define in non-humans. But what there were more robust findings for was anxiety. And
Hmm.
anxiety, it seems like not only in humans, but across species and beginning at puberty is higher in females. And I was wondering if you know of any of the research on that and like what the hormonal mechanisms are, like what ovarian hormones are doing and why anxiety or depression, internalizing symptoms more generally would go up.
Short answer is no. Yeah, I just
I mean,
haven't,
no, they don't, or no, I don't know enough.
I don't know it. Yeah, I just haven't reviewed that literature. But yeah, I mean, it's an interesting pattern. And when you see a robust sex difference, some involvement of sex hormones, either organizationally or
Oh yeah,
actually
there was one more layer
jumps
of
toward.
complexity that I missed, which is that it might not be that there's a positive anxiety boost in females. It could just be that testosterone inhibits anxiety, which I've seen research on.
Yeah, I guess the only thing I really know about that is there are some findings looking at cycle phase shifts. And the general gist is that near ovulation in the fertile window, which is the time when estradiol is higher, progesterone lower, there's also a testosterone elevation, a modest testosterone elevation at that time. But generally, women report feeling better, higher. Most of the evidence suggests higher self-esteem, feel more attractive.
And that's
Some
linked to the
evidence,
testosterone.
no one's shown that it's linked to testosterone, but just time in the cycle as just a clue to what you were just referring to. So yeah, it just seems to be a time when other things equal, of course, and the hormones and the time in the cycle are probably accounting for way less variance than just things that are happening in your life on that day. But yeah, that's the only pattern that I am confident in with respect to anxiety. And I don't even know that it's anxiety per se, just things that are
Mhm.
adjacent to anxiety. So better mood, better self-esteem.
Right.
So
Is it true that holding other things constant, higher testosterone is associated with higher self-esteem, more motivation, more confidence, more dominance?
so baseline testosterone or a reactive increase. And
I guess either. I could think of three ideas. So one would be the reactive change that you mentioned. One would be baseline levels, and then one would be something like more of a long-term organizational effect of like higher testosterone levels over the course of development can lead to brain changes that indirectly lead to this stuff.
I think in general, personality correlations at baseline with testosterone are pretty modest and they have kind of conflicting results as to whether you get significant effects or not. It's usually based on a single sample or two samples and then a correlation with some measure. So any sort of self-report behavioral psychological measure, generally the correlations are pretty modest and even inconsistent. There is some evidence for reactive testosterone increases being associated with more reactive aggression, willingness to compete, increased competitiveness, maybe increased confidence, reduced fear responses like fear, startle responses. So just a suite of things that are related to competitiveness and might also be related to things like courtship effort. I actually forgot the specific variable you first started this with.
I wasn't that specific with it. I threw out dominance, confidence, self-esteem, all in this general, assuming they're associated.
Yeah. So those just the designs are there's a mix of designs. So there are things where you do something to a subject, measure their testosterone change, a reactive short-term change, and then measure what's correlated with that change. And then more rarely there are studies where they actually administer testosterone and then look at outcomes. So in the ones where they trigger changes, yeah, there have been studies where when testosterone went up after a manipulation and then they put men together with women, the men were more likely to talk to the women, approach them, and so on, when if they had a larger testosterone increase. Interpreting causation there is a little difficult, right? Because it's not like you've isolated just testosterone in that case. Whereas when they give subjects testosterone, yeah, I don't know if confidence per se has been directly measured, but things like, yeah, reduced startle responses, fear responses, things that are at least adjacent to confidence. So the argument is have increased.
Yeah, I was talking to a friend and peer of mine, hi Rachel, about this recently, about sex differences in aggression and sex drive and anxiety, and telling her what I know about how it might be linked to testosterone, whether that's circulating levels or prenatal organizational effects during puberty. And we seem to accept that this main effect of sex difference might be largely explained by hormones. But then the question was, what about individual differences? And we already talked about that, talked about whether continuous differences in, say, testosterone does in fact predict aggression or sex drive. And it seems like maybe not if it's in a broad enough range. But she asked, what about women who are hypermasculine? So women who are more aggressive or who have higher sex drive or more tomboyish interests, is there any link to testosterone levels? Again, whether it's on the continuous end or whether it's due to organizational effects early in development. Is- I know a little bit about this in the context of CAH. So girls with CAH, but they would have much elevated testosterone levels. So it wouldn't just be like at the high end of female normal. It would be like way beyond that up into the male range. But there's a bunch of studies on children with CAH, girls have more rough and tumble play preferences. They're more aggressive. They're more tomboyish. And I was wondering if within the normal female range, at the high end of it, there's anything that could be predicted there.
Yeah, not that I know of. So again, my focus hasn't, I did a little bit of the CAG repeat work on individual differences, but for the most part, yeah, I haven't been focused on individual differences very much in the research, trying to discover the basic typical mechanisms, the main effects first. But yeah, my rule of thumb just from the last time I reviewed the baseline testosterone literature, there could be some new developments there, but is that, yeah, if you just measure someone's baseline circulating hormones and try to correlate it with whatever it might be, including things like physical strength and then aggressiveness, personality traits, you tend to get really small, modest correlations. And so part of that, again, as you're alluding to, could be that a lot of the action was already the earlier hormone exposure and the organizational effects, including organizational effects of puberty. So not just prenatally. And then those individual differences have been calibrated before you're measuring the current hormones. And hormones are going up and down for functional reasons, and you're just getting a snapshot of them. Then you don't necessarily expect to see a really large correlation between that one hormone measurement and something like personality traits or tendencies toward more masculine or feminine sort of interests. And so,
Jim, it sounds like much of your recent research has been on these daily diary studies, and that the future of the field is looking towards more short-term, continuous within person changes and maybe interactions between these different hormones. What are you currently working on, and what are you most excited about?
yeah, a number of things. So yeah, I am still trying to come up with an overall model of what hormones are doing in women's sexual motivation, which I think is an understudied topic in some ways.
Do you work with
So
Emily Jacobs at all?
a little bit, yeah, when we talked about collaborating more, it's always just getting enough time when you're
Mhm.
juggling everything. But yeah, no, her lab's done some really interesting work. And right, she more on we were talking about earlier, there's these different levels of analysis. I think she's
Mhm.
closer to the brain region, cortical connectivity, and so on, estradiol across the cycle. Yeah, I'm still trying to develop an overall model of how motivation shifts across the cycle and the role of hormones in regulating that. And yeah, we're finishing another really large daily diary study. We're trying to tease apart some of the remaining ambiguities in that literature and trying to replicate some of the prior findings. So that's one line of research. There's another line of research where we're looking at concealed ovulatory timing. Are there cues of when women are near ovulation, fertile window, and what's the role of hormones in that? So we published a couple of recent papers on that, looking at odor attractiveness across the cycle, how it fluctuates, and how hormones might be associated with that.
How does
And
that work with smell and with pheromones? Because I hear the word pheromones a lot in animal research, and especially when you're talking about these effects for mating, like during ovulation, maybe the female releases certain pheromones and the males smell them, and then that's what triggers their sexual response. But I've heard that humans don't have pheromones, but I've also seen all these interesting studies looking at sweat stains from males are perceived as more attractive when women are ovulating, or conversely, men will find the sweat of ovulating women more attractive than non-ovulating women.
yeah, it's the latter that we were looking at. Do women smell better near ovulation? And so first there's this question of does it would any such signal or any such cue meet the definition of a pheromone? And from my perspective, I don't really care about what the technical definition of pheromone is.
But what is
People
the
argue.
technical definition? Because I was thinking about it as they're more or less the same chemicals, but hormones are in your blood and pheromones are in the air.
I don't know what the technical definition is because it doesn't matter to me, essentially. So people have published articles saying it must meet these 7 conditions or it's not technically a pheromone. So the only thing I want to know is, are the odors different? Do they carry information that perceivers could use essentially to detect ovulatory timing? And so what we did in our study we recently published it's a logistically difficult study, but another daily diary study. But every 5 days, we had women collect odor samples overnight. So we would launder T-shirts and give them to them. We're running a laundry in our lab, and so on. And so they would collect these samples overnight. And then we were also getting saliva samples to measure their hormones on the same days and using these urinary luteinizing hormone tests to detect when they ovulated. So we're getting these 6 different samples spread across the cycle with their hormones, and we know where they are in the cycle, in the ovulatory cycles. And then we had brought in men, put the samples in these glass jars, and the men would sniff them and rate them for attractiveness. And we did see a significant increase in ratings of attractiveness for samples that were in the fertile window, estimated fertile window, relative to other regions of the cycle for the same women. So we're doing this within woman comparison. So on average, they were rated as smelling better. But one of the things that we did that was original in this study is we applied signal detection theory to try to assess whether men could really diagnose fertile window timing from those attractiveness ratings. And so the basic idea there is that we're assessing the degree of overlap between samples that were from the fertile window, their attractiveness ratings, versus samples that were outside of the fertile window. So even though there's an on average difference in odor attractiveness, what we found was that there were so many samples outside the fertile window that overlapped in ratings from those inside the fertile window that there's basically no diagnostic information at all. So the signal detection analysis suggested that despite a significant effect, there was really no ability to detect ovulatory timing.
So within people, you could detect a difference, but then just like sampling from the population, you have no idea if you're really attracted to this one because of the slight increase due to ovulation or just due to a whole bunch of other factors that could make one woman smell more attractive than another. So like some non-ovulating woman would still smell better than the ovulating woman, even if within each woman you'd see a difference, like across woman you wouldn't.
That is contributing to what I'm saying. But actually, even when we set up the signal detection analysis so that we were comparing within women, what we found was that, so for a given woman, maybe on average, her samples are being from the fertile window, that fertile window sample is being rated higher than the others overall on average. But there would still be samples that overlapped, would be rated as high as her fertile window sample. So even within the same woman, we found low detectability when we set up the analyses that way. So what you had pointed out is really important, that some women, there are between women differences. Some women just smell better than others. So even if they're outside the fertile window, they smell better than a woman who's inside the fertile window. We definitely find that, right? And so that suggests if you're just smelling strangers, you're going to have no idea who's in the fertile window. But in our other analyses, we also found that even if we compare the same women's samples and applied the signal detection analyses, that detectability was still very poor, even in that case. So even when you're comparing 6 samples from the same woman, even if the fertile window sample on average does a little bit better, you're going to have in most cases, there are going to be other samples that are rated just as high out of those 6. And so you get too much overlap to really have a good diagnostic view of whether they're in the fertile window or not.
Does this mean your theoretical framework is heading more in the direction of whatever the effects of hormones are, they're more individual specific? So it's not really about detecting other people's hormone levels. It's more just about what your hormone levels are doing to you and your perceptions.
Not sure I fully understand that. The conclusion we're drawing is that there might be like these little, very subtle leaking of some cues that may slightly increase odor attractiveness, but there's so much overlap with the non-fertile samples that from odor, at least, ovulatory timing is effectively concealed. So the question we're addressing is, okay, do we have concealed ovulation or not? And I think we do, in spite of there being a significant effect, which is the subtle part of the argument.
Yet another interesting paradox.
Yeah. Yeah. And then the other thing we did was we correlated the hormones with the odor attractiveness. And within women, we didn't see significant results. And what we were theorizing was that if it were possible to detect ovulatory timing, you would expect that progesterone would be negatively correlated with odor attractiveness because progesterone is only going to rise when women are not fertile window. The fertile window is passed or they're pregnant. So progesterone body odors associated with high progesterone days should smell bad if perceivers can detect fertile window timing from odors, but they didn't. The correlation with progesterone was essentially zero within women. And so to us, we were theorizing that, so in certain non-human species, it's clearly the case that females do signal their ovulatory timing and males respond to it. And so we were theorizing that the suppression of progesterone effects on cues of ovulatory timing might be part of the kind of proximate mechanism for concealed ovulation in humans. So we call that the progesterone suppression hypothesis. Try to break that link between progesterone and observable cues. But it's one study, right? So we need to follow up.
That's very interesting. I'm looking forward to seeing more about what comes out of your lab.
Yeah, great.
Thank you very much for your time, Jim.
Yeah, my pleasure.